You have roughly 38 trillion bacteria living inside your gut right now. They are not passive passengers. They respond to every meal, every fast, every hour you go without food. And when you push that window to 72 hours, something inside your intestines shifts in ways that scientists are only beginning to fully understand. What exactly happens to your gut microbiome when autophagy kicks in and your body starts cleaning house at the cellular level? That is what this video is about. The human gut is not simply a digestive tube. It is a densely populated ecosystem hosting somewhere between 500 and 1,000 distinct bacterial species, according to research published in Nature by Dr. Rob Knight and colleagues. The collective genetic material of these microorganisms, what scientists call the gut microbiome, contains roughly 150 times more genes than the entire human genome. That number alone reframes how we think about the body. What makes this ecosystem particularly relevant to a conversation about fasting and autophagy is that the gut lining itself is one of the most metabolically active surfaces in the human body. The intestinal epithelium renews itself every 3 to 5 days, and the cells responsible for that renewal, intestinal epithelial cells, are among the first to engage autophagic processes during periods of nutrient deprivation, as described in research by Dr. Ana Maria Cuervo at Albert Einstein College of Medicine. The gut is not just hosting bacteria, it is actively interacting with them at the cellular level. This interaction runs deeper than most people realize. Gut bacteria produce short-chain fatty acids, neurotransmitter precursors, and immune-modulating compounds that influence everything from intestinal wall integrity to systemic inflammation. When the nutritional environment changes, as it does during a prolonged fast, these microbial outputs shift accordingly. Understanding that baseline is what makes the 72-hour window so scientifically interesting. In the first 12 hours of fasting, the body is still largely running on glucose derived from glycogen stores in the liver and skeletal muscle. From the gut's perspective, the most immediate change is mechanical. The migrating motor complex, a pattern of muscular contractions that sweeps the intestine clean between meals, becomes the dominant motility pattern. This housekeeping wave, which is suppressed every time food enters the digestive tract, now runs uninterrupted. At the microbial level, bacteria that depend heavily on dietary sugars and simple carbohydrates begin experiencing reduced substrate availability. Species within the Bifidobacterium genus, which ferment dietary fibers and simple sugars, start competing for remaining residues in the colon. A 2019 study in Cell Host and Microbe noted that microbial composition begins showing measurable shifts within the first 10 to 16 hours of caloric restriction, even before ketosis is fully established. The intestinal immune system also starts recalibrating during this window. Secretory IgA, the primary antibody class that lines the gut mucosa, maintains relatively stable levels in early fasting, but the inflammatory signaling environment begins to quiet. Research from Dr. Laura Hooper's lab at UT Southwestern has documented that the relationship between intestinal epithelial cells and resident bacteria is exquisitely sensitive to nutrient status, and the first 12 hours mark the beginning of a measurable immunological recalibration. Between hours 16 and 24, glycogen reserves are substantially depleted in most individuals, and the liver begins ramping up ketone production. This metabolic transition has a direct effect on the gut environment. As glucose availability drops, the luminal pH in parts of the colon shifts slightly, and this change in chemical environment begins selecting for different bacterial populations than those favored in a fed state. This is also the window in which autophagy begins operating at a more significant scale in gut epithelial cells. Dr. Beth Levine's foundational research at UT Southwestern, including work published in Science, established that nutrient deprivation is one of the primary triggers for autophagic activation. Inside intestinal epithelial cells, the mTOR pathway, which normally suppresses autophagy when nutrients are abundant, begins losing its inhibitory grip, allowing autophagosomes to form and begin engulfing damaged organelles and misfolded proteins within the gut lining itself. What happens at the mucosal interface during this period is particularly striking. The mucus layer that coats the intestinal wall, produced by goblet cells, is a primary habitat for bacteria like Akkermansia muciniphila, a species extensively studied by Dr. Patrice Canny at UCLouvain for its association with gut barrier integrity and metabolic health. During fasting, Akkermansia has been observed to increase in relative abundance in animal models, partly because it can degrade mucus as an alternative energy source when luminal nutrients diminish. Whether this represents a beneficial adaptation or a stress response to mucosal depletion is still an active area of research. Not all gut bacteria respond to fasting the same way. The microbial community inside the colon is stratified by metabolic strategy, and a 72-hour fast functions like an ecological pressure test. Species with flexible metabolic repertoires tend to hold their ground or expand, while those dependent on a constant stream of dietary input contract sharply. A 2020 study in Nature Microbiology examining prolonged caloric restriction in humans documented significant reductions in Prevotella species, which are heavily dependent on dietary carbohydrates for fermentation substrate. On the other side of that equation, bacteria capable of cross-feeding, meaning they can survive on metabolic byproducts produced by other microbes rather than directly on dietary input, tend to persist or increase. Faecalibacterium prausnitzii, one of the most abundant butyrate-producing bacteria in the healthy human gut and extensively studied by researcher Harry Sokol at Sorbonne University, shows a complex response to fasting that appears linked to the availability of acetate and lactate produced by other bacterial species. Butyrate itself is the primary energy source for colonocytes, the cells lining the colon, which creates a feedback loop between microbial activity and epithelial cell survival during nutrient scarcity. Akkermansia muciniphila continues its relative expansion into this window, and researchers have proposed that its increase during fasting may serve a structural function. By degrading and stimulating the renewal of the mucus layer, it may actually promote goblet cell activity and mucin replenishment rather than simply depleting the barrier. A 2017 paper in Gut described how Akkermansia abundance correlates inversely with intestinal permeability markers, a finding that gives its fasting-associated expansion particular biological significance. By the time a fast crosses the 24-hour mark and moves toward 48 hours, autophagic flux in intestinal epithelial cells is operating at a substantially elevated rate. The term autophagic flux refers not just to the initiation of autophagy, but to the complete cycle, the formation of the autophagosome, its fusion with the lysosome, and the successful degradation and recycling of the engulfed material. Dr. Daniel Kliansky at the University of Michigan, whose laboratory has produced some of the most cited methodology in autophagy research, has emphasized that measuring flux rather than simply autophagosome accumulation is critical to understanding whether the process is actually functional. Inside the gut lining specifically, autophagy serves functions that go beyond simple cellular housekeeping. Paneth cells, specialized secretory cells located at the base of intestinal crypts, are among the most autophagy-dependent cell types in the body. These cells produce antimicrobial peptides that regulate bacterial populations in direct contact with the epithelium. Research published in Nature by Dr. Herbert Virgin at Washington University demonstrated that defects in Paneth cell autophagy are associated with dysregulated microbial communities and increased susceptibility to intestinal inflammation. During a prolonged fast, the autophagic activation of Paneth cells may represent a form of quality control that maintains the precision of antimicrobial secretion even under metabolic stress. The intestinal stem cells residing in the crypts of Leberkuhn also engage autophagy during fasting in ways that appear to influence their regenerative capacity. A landmark 2018 study in Nature from Dr. Omer Yilmaz's laboratory at MIT showed that fasting enhances intestinal stem cell function through a fatty acid oxidation pathway, and that this enhancement was linked to autophagic activity within the stem cell niche. The implication, still being explored, is that the gut lining's capacity to renew itself may be influenced by the autophagic state of its own stem cell population. The gut and the brain maintain a continuous bidirectional communication network, the gut-brain axis, that operates through neural, endocrine, and immune channels simultaneously. During a 72-hour fast, this communication system does not go dormant. It shifts register entirely. The vagus nerve, which carries sensory information from the gut to the brainstem, begins transmitting a different pattern of signals as luminal content changes. Intestinal motility patterns shift, and microbial metabolite production reorganizes. One of the most consequential microbial contributions to this axis involves the production of short-chain fatty acids and their influence on enteroendocrine cells, specialized gut epithelial cells that sense the luminal environment and release hormones accordingly. During fasting, as bacterial fermentation activity changes, the profile of gut-derived signaling molecules reaching the bloodstream and the vagus nerve shifts. Research from Dr. John Cryan at University College Cork, one of the leading figures in gut-brain axis research, has documented that microbial metabolites including butyrate, propionate, and specific amino acid derivatives influence serotonin synthesis in the gut, where approximately 90% of the body's serotonin is produced. Tryptophan metabolism is particularly relevant here. Gut bacteria from the Lactobacillus genus and others influence the conversion of dietary tryptophan into either serotonin precursors or kynurenine pathway metabolites, a balance that has implications for both intestinal function and neurological signaling. During extended fasting, as dietary tryptophan input disappears and microbial populations shift, this metabolic balance reorganizes. A 2021 review in Cell by researchers at the Broad Institute described the tryptophan-kynurenine axis as one of the most dynamically responsive microbial host interfaces during periods of nutritional change, a finding that adds a neurochemical dimension to what might otherwise seem like a purely digestive event. Chronic low-grade intestinal inflammation is one of the most studied and most misunderstood phenomena in modern gastroenterology. The gut immune system is in a perpetual state of controlled activation, managing an interface where trillions of bacteria live in proximity to the bloodstream. What happens to that inflammatory tone after 72 hours of fasting is not a simple on-off story. It is a recalibration of multiple overlapping systems happening at different speeds. One of the most documented changes involves NF-κB signaling, the transcription factor pathway that drives the production of pro-inflammatory cytokines, including TNF-alpha, IL-6, and IL-1 beta. Research published in Cell Metabolism has shown that caloric restriction and fasting suppress NF-κB activity in intestinal epithelial cells, and that this suppression is partly mediated through the same AMPK pathway that activates autophagy. In other words, the metabolic signal that turns on cellular cleanup also turns down a key driver of intestinal inflammation. These are not separate events, but interconnected responses to the same nutritional state. The role of butyrate in this context deserves specific attention. As Faecalibacterium prausnitzii and other butyrate producers maintain activity during the fast, feeding on cross-fed substrates rather than dietary fiber, the butyrate they produce continues reaching colonocytes and exerting its well-documented anti-inflammatory effects through histone deacetylase inhibition. A 2019 paper in Nature Immunology from researchers at the Garvan Institute described how butyrate directly influences the differentiation of regulatory T cells in the gut, immune cells that suppress excessive inflammatory responses. The microbial contribution to intestinal immune regulation does not simply stop when dietary input stops. The idea that prolonged fasting can reset the gut microbiome has circulated widely in popular health media, often with a level of certainty that the actual science does not support. The honest answer is more nuanced and more interesting. What the research shows is not a wholesale replacement of microbial populations, but a temporary and partially reversible reorganization of community structure that depends heavily on what comes after the fast. A 2019 study in Cell by Dr. Eran Segal and Dr. Aaron Elinoff at the Weizmann Institute of Science examined gut microbiome dynamics during and after the Ramadan fast, a model of intermittent daily fasting over 30 days, and found that microbial shifts during fasting were largely reversed when normal eating resumed, with the trajectory of recovery shaped significantly by the quality and composition of the refeeding diet. A separate study published in Gut Microbes examining water-only fasting found that while alpha diversity, the variety of species within an individual's microbiome, temporarily decreased during extended fasting, certain keystone species showed durable changes in relative abundance that persisted beyond the immediate refeeding window. Dr. Justin Sonnenberg at Stanford, whose research on microbiome plasticity has been published extensively in Nature and Cell, has argued that the microbiome is far more dynamic than previously believed, capable of meaningful compositional shifts over days rather than months. That dynamism is precisely what makes the 72-hour fasting window biologically significant from a microbial standpoint. Whether that significance translates into long-term ecological change in the gut remains one of the genuinely open questions in the field. When food is reintroduced after 72 hours of fasting, the gut does not simply resume where it left off. The refeeding window represents a period of heightened microbial responsiveness, a state in which the composition of what enters the gut has an outsized influence on which bacterial populations expand first and how quickly. Research from Dr. Aaron Elinoff's group at the Weizmann Institute has specifically characterized the post-fast gut as a state of elevated microbiome plasticity, where bacterial communities are more susceptible to compositional change than they are during habitual eating patterns. The intestinal epithelium itself undergoes rapid structural recovery during refeeding. The tight junctions between epithelial cells, proteins like claudin and occludin that determine intestinal permeability, begin restoring their configuration as nutrient-signaling resumes. A 2020 study in mucosal immunology documented that the reintroduction of specific fermentable substrates after a period of fasting preferentially expanded Akkermansia, muciniphila, and Faecalibacterium prausnitzii before other species recovered their pre-fast abundances, suggesting that the early refeeding environment shapes the trajectory of microbial recovery in a sequence-dependent manner. The autophagic machinery also transitions during this window. As mTOR signaling resumes with nutrient availability, autophagy returns to basal levels, but the cellular quality improvements achieved during the fasting period, including the clearance of damaged mitochondria and misfolded proteins from gut epithelial cells, persist. Dr. Ana Maria Cuervo's work on autophagic efficiency has consistently shown that the benefits of a completed autophagic cycle are not reversed simply by refeeding. The cellular landscape of the gut lining after a 72-hour fast and refeeding is measurably different from what it was before the fast began. The scientific picture that emerges from 72 hours of fasting and autophagy in the gut is not a story about one mechanism or one cell type. It is a story about interconnection between the microbiome and the epithelium, between autophagy and immune regulation, between microbial chemistry and neurological signaling. What makes this research frontier genuinely exciting to scientists is precisely that complexity. No single variable explains the observed changes, and that means the field is still in early stages of mapping a system of remarkable biological depth. Some of the most promising current research directions involve using the fasting state as a controlled experimental window to study gut biology with unusual precision. When dietary input is removed, the confounding variables introduced by food composition, meal timing, and digestive activity are temporarily eliminated, giving researchers a cleaner signal for studying baseline microbial behavior, epithelial cell dynamics, and autophagic flux. Laboratories including those of Dr. Sarkis Mazmanian at Caltech and Dr. Wendy Garrett at Harvard School of Public Health are actively investigating how periods of nutritional stress shape long-term microbiome stability and host microbe co-regulation. What 72 hours of autophagy ultimately reveals about the gut is that the body's interior ecology is not static. It is continuously negotiating between microbial demands, epithelial maintenance, immune surveillance, and metabolic state. And fasting compresses that negotiation into a window that science is only now equipped to observe with the resolution it deserves. The researchers working on these questions are not just studying fasting. They are building a new map of how the human gut maintains itself, and what happens when the environment it evolved in is temporarily, deliberately removed. If you found the science in this video worth your time, consider subscribing to the channel. Every video here is built on published research and explained without shortcuts. Share this one with someone who thinks gut health is just about probiotics. And if you have a question about anything covered here, the comment section is the right place for it.